All right, good morning, everybody. Welcome to the 2020 BF MS webinar series. Glad to have you here today. Thanks for taking some time out of your busy day. I know it's harvest for a lot of people right now. Kind of nice, maybe with the heat, at least we're having in the Pacific Northwest, to be inside, and maybe the rain on the East Coast. So anyway, I want to get started today. Did want to let people know that Jenny is taking some needed time off today. We miss her today, but she is not here. So unfortunately, I have Steve running the operation behind the scenes. So this could be trouble. But what I do want to, I want to let everybody know that as we go through the presentation today, if you have any questions, please type those questions in and send them. I really like to try and answer questions when we're still on the same topic that the question pertains to. Questions are wonderful for me. It gives me a chance to learn of what information and questions growers have out there today. It gives me a chance to educate myself on things that I need to learn or better understand. And some of the questions we can answer, and some of them we'll definitely research and find an answer for you. So please feel free to send those in. Steve will try and monitor those as we go through on the webinar here. So today, Steve and I would like to talk to you a little bit about why biology. And, you know, I look at that in a little bit different than what we've talked about in the past. We're going to go beyond just, you know, soil health will be part of it, nutrient availability will be part of it, but we're going to talk a little bit more about why biology and really how important biology is. And there's so much wonderful new information coming out today. We'll talk a little bit about how our products pertain to that information and how we can utilize our products in the best way. We get a lot of questions here. We've got quite a few products out there and which one makes the most sense. And then we want to spend a little bit of time on what's new. There's some really cool new research coming out that we want to just touch base on that will be wonderful tools for the future for us to use as growers to better apply biology to what we're actually doing. And then we'll get in a little bit to questions. So with that, Steve, you want to say hello?
Hello, everybody. Thanks again for joining. As Dennis has already said, I'm going to have lots of fun, cover a wide variety of topics, and hopefully everybody finds something new, something entertaining. And like Dennis said, share those questions. We love to have those questions because it does help guide where we go and what we talk about. So thank you.
Yep. So let's get started. So with that being said, you know, I like this quote from Dr. Jerry Hatfield: soil biology and biological fertility is the key to future of agriculture. And Dr. Hatfield has probably been talking about this and researching this for at least 6 or 7 years, if not longer. And a lot of what he has taken a look at is how important the entire biological system is to the future of agriculture. And I want to tie this in pretty much in the first part of this presentation of some things that we'll talk about of really how everything is connected and how important it is. And it kind of goes back to the story. You can see that beautiful rooster pheasant. That was a picture that I had taken. This had happened on one of my growers in Colorado. And the reason for this, I was working with this grower and he and I were having a conversation because he knew my love of pheasants and my understanding of pheasants and what needed to happen in order to bring pheasant population back onto his ranch. And the reason for that is he wanted to do this in memory of his son, because he had fond memories growing up during the early season of October and hunting season of his son going out on the hay fields and shooting pheasants and bringing them back and having them for dinner. And, you know, probably 15 or 20 years ago, they changed their practices to where they weren't so much a cattle operation. They got rid of most of their cattle and they were mainly producing hay. And so they went into their pasture ground, which was predominantly just grazed. They took out all their fences. They basically went into hay ground. So they started to change their agricultural practices. So they went into spraying herbicide to control the weeds, spraying insecticide to control the insects, basically cutting their fields. On this, they were trying to get 3 cuts a year. Mainly, they got 2 and a little bit left over on their late cut. It wasn't great. But he noticed that when they made that transition, slowly the native pheasant population went away and they no longer had pheasants. And so we started to talk a little bit about that. And it really ties into agricultural practices. And as I told him, what is happening was the herbicides and the insecticides were being sprayed during the nesting time of the pheasant. And one of the things that is critically important to young pheasants is insects. That's their main food source when they're young. They had also taken out all of the habitat, the weed piles, the brush piles, everything that were native in these pasture grounds for cover or homes or protection for these pheasants. So when they would go in and hay the ground and cut everything off, these pheasants had nowhere to hide. They had no homes, they had no protection from environmental influence and environmental stress. And so slowly we started to see that pheasant population basically just disappear. There were a few left here and there, but nothing like he had remembered. It was a rare sight for him to see a pheasant. So we went in and we kind of changed what he was doing. He went into more biological fertility programs. He went away from most of his weed application, at least blanket spraying across the entire farm. He went to spot spraying. He let some of these native brush patches grow back up. And the reason for that is because when he mowed off the field, the pheasants had somewhere to go for protection from the environment, from environmental stress. And the other thing that he started to take a look at is that last cutting in the year, he no longer mowed. There wasn't really enough hay that he was taking off of that ground. And so he left that as basically habitat for those birds for over the winter time. And what we did is we came in and we released 50 roosters and 50 hens that first year. And then since then, about every year, he'll put out about 25 hens onto this just as brood stock to help reproduce, to keep that population going based on owls, hawks, all the predators, all the things we see in agriculture that we basically disrupt that environment. And this is really true what we have to do when we start to look at increasing our biology within our soil environment. Now, the difference here is with my understanding of pheasants and where pheasants like to be, what pheasants need for food, what they need for habitat, what they need for homes and water, and what their food source is and all those things, I can visually stand on that property, look across that property and see what's missing. In agriculture, it's not that easy. We generally take a look at our crops. We look at them based on nutrition. We say, okay, my plant needs water, my plant needs fertilizer in order to grow, but I can't see what is actually happening or missing within that biological community, within that soil environment. So we kind of have to do research and get an understanding of our practices. Like I was saying, the herbicide use, the insecticide use was a direct food source for those pheasants. So we have to start tearing agriculture apart and looking at the whole full picture as we go back in to try and building soil health. And then when we can reintroduce our microbial population and supplement that population year after year, and we can actually have a thriving population of biology back into that soil environment. And I kind of touch on this a little bit because this all came about to me based on reading an article when I was doing a little research 2 weeks ago. And it was an article by Cornell University and Washington State University. And I thought, well, this sounds kind of interesting. And the title of it was Farmers' Climate Change Conundrum, Low Yields or Revenue Instability. And what they're talking about here, the main focus of this article is drought. We have less water, we have more stress, we have higher temperatures, and it is leading to crop failure or crop yield reduction. And the whole basis of this article was going into producing seed hybrids or genetically modified seed or changing seed varieties to handle stress better and lower moisture rates, more drought stress. And I started to think about this. Nothing in the entire article talked about soil health. Nothing of it talked about nutrition. Nothing of it talked about things that we have been talking about with the biological farm management system, for the last 30 years. And it kind of made me think back to 30 years ago when Bruce started this company, over 30 years ago, 35 years ago. Bruce was producing hybrid seeds, and he was trying to produce better seeds to produce better crops. And 30 years ago, he figured out he was headed in the wrong direction. By just changing the crop and not changing the environment in which we grew that crop in, we were not actually changing the outcome of what we were really trying to control. Because environment is, it's tough. It's the one thing that we can't control is the weather. And so what we need to do is do everything in our power to reduce the stress that is brought on to us by Mother Nature and by that weather. And it's not always just changing seed variety. Seed variety helps, but we have to look at overall soil health. We have to look at that biological community. We talk so much about building a microbial community, building soil carbon, building soil structure. As Steve puts it, building a bigger sponge in order to hold more water, to reduce that stress on that plant. And not only by having better water holding capability, but better nutrient holding capability, and paying attention to how we feed that plant. And so that was really what I want us to start thinking about today as we go through this presentation of what things can we do to basically swing things to our advantage so we no longer have to look at these low yields or revenue instability. Because what I look at is anytime we create a seed hybrid, that actually increases the cost to the grower for a more expensive seed. It's a better quality seed. When it first comes out, it's going to be more expensive. Let's look at putting that same money of what that seed cost would be, maybe into building soil health, and try and work at it in a whole different direction, as Bruce used to talk about and has been talking about for the last 35 years, and we continue to talk about today. And that was part of the reason why we talk about the biological farm management system. Steve and I always talk is it's not a product, it's a program. And so with that being said, Bruce knew that a long time ago. It's a system. It's a biological farm management system. Everything has to come together in order for this to function properly.
Well, and a big part of that and what we like to see, like you talked about with the pheasants, as they switched over to some of their practices and reduce some of the diversity on that field, that automatically has an impact on other species there. So a big part of what we try to do is emphasize and maximize the species diversity. And a big part of that, it's not just plant diversity. That can also be insect diversity. It can be bacterial, fungal population diversity, because each one of those players has a part. It does play a part to the overall picture. It's kind of like puzzle pieces where you put them all together, and once they're all together, they can actually form that full structure, that full picture. And the biological benefits that we see can be pretty incredible, and we have such a wide diversity of organisms. And as we research and study the plant microbiome, we see more and more how critical these microbes are to how that plant functions. So Dennis was just talking about changing genetics, changing to different hybrids. And a big part of that is focusing on the genetic diversity, or, reducing diversity in some cases of these seedlines. That ignores the microbiome completely. And as we remember and as we think back, like in a human, we're only 10% human DNA. There are 10 trillion human cells and 100 trillion bacterial cells, 30,000 human genes, 1 million bacterial genes that are making our bodies function the way they are. And these plants are the same. So if we unlock the potential of what these benefits of microbes can do, we can see benefits across the board.
And that's a really good point, Steve, and kind of brings into exactly what I want to talk about here in the next slide is, as I say, the benefits of biology. But to receive the benefits of biology, we have to think about where biology is. And, so often we talk about the rhizosphere and the phyllosphere. We talk about surrounding the roots and surrounding the leaves. But what we're missing here and what we don't talk about enough, and this is kind of why I wanted to chat about this a little bit, is we don't only have it surrounding the roots. We don't only have it in the soil. We don't only have it on the root surface. We have it on the flowers. It's on the fruit. It's on the stem. It's in the leaves. It's within the tissue of the plant. It's surrounding the seed. You know, biology is everywhere. There's a lot of research out now coming on how the biological community changes when grapes ripen, how it changes on olives. And that's not only true, the research is going there, but that's true on every crop that is grown. And we have to really pay attention to that. You know, I was talking to a grower. I was up in Tonasket last Thursday talking to some growers. And as we were walking through this orchard with one of the growers, and I drove into this orchard, and to me, visually from the distance, the orchard looked really good. And we went in there and I was walking through the orchard and we were chatting. And one of the comments that was made to me was, the orchard looked so amazing in the spring. And she said, it just doesn't look as good now. And I said, well, what did we do differently? And she said, well, you know, we started to spray our lime sulfur. We started to spray our dormant oils for coddling moth. We started to do all of our control products. And if you think about it, it goes right back into what we're talking about here. We changed the microbiome on that plant by spraying those things. And as long as we know that, as I always say, now we can go back and do something to fix it, okay? Because as we just talked about, we have biology on the flower, on the stem, on the leaves. When we suffocate them with lime sulfur or copper fungicides or dormant oils, all of these things is changing. And so we need now to go in and fix it. That's what we talk about. That's how we try and manipulate our environment based on things we have to do in agriculture in order to be successful. So, you know, in the springtime, I talked to a lot of my growers about foliar nutrition is so important to bud swell. And if you think about that, a lot of times I get the question, well, there are no leaves out there. How does foliar nutrition help? But when we start to think about biology and nutrition in that plant, it starts long before we have leaves. As we have bud swell, we're starting to set flower, we're starting to set this year's fruit, which has already been determined starting way back last year. But there's a biological community everywhere, on the stem, on the bark, on the leaf, in the plant. So everything we can do to feed that is going to enhance the overall health of our plant. And those are some of the things that we just want to kind of keep up with today and follow.
Well, in a conversation we had here pretty recently, Dennis, we were kind of talking about the idea of control. When we're using a control product, we're purposely trying to manipulate. We're purposely trying to control the environment, to change a function of what's going on in the plant. And sometimes that's absolutely necessary. If it comes down to having a crop or not having a crop because we're being devastated, we have to try to take that into hand. We have to try to control that situation. But we also need to remember, we need to go a step behind that of why did we end up here? And as you were just talking about, as we apply these materials, we're having unintended consequences. And the beauty is, like you said, Dennis, we have ways of overcoming that. It's knowing that I'm doing a little bit of damage. If I know that I'm doing a little bit of damage, then I can do a little bit of preventative maintenance and help get that system back up and running. And that's what a lot of these microbes do. And as we research more and more, we can see more and more of their functions. And some fairly recent research is really highlighting how incredibly critical some of these systems can be.
You know, and it goes right into what you talked about. I believe it was last week or maybe it was in March. And we talked about, if any of you out there haven't done a little bit of research or Google Dr. White, I would highly encourage it. It kind of brings this to the next level. Some great information out there. And Steve has talked about the rhizophagy cycle. Sorry, that's really hard for me to say. But it goes back and it emphasizes what we're talking about here is how this biological community, these microbes, enter into the plant, into the root. That root then takes them, digests them, and spits them back out into the soil, where they now basically are hungry. They need to rebuild their bodies. They feed, they basically get their health back up, and the cycle starts all over again. And then that plant mines this nutrition out of them. And it's just an ongoing cycle. That's not only in the root. We're finding that happens in other parts of the plant. It happens on the leaves. So when we start to talk about that, we need to really think about this goes really to the next level and how this system works. And again, as I talked about with the pheasants, we can't see that standing looking across and finding out what we need for the pheasants in order to make that population grow. All of this happens naturally within that soil environment. And anything we do in our agricultural practices to disrupt that, which there are things that we have to do, we have to then step back and say, okay, this is what I have done. What do I need to do to fix it? Or what tools do I have? A lot of times that's re-inoculation. A lot of times that may just be feed. A lot of times that may be getting a cover crop in there and establishing a good cover crop with a good healthy microbiome. There's a lot of tools that we have out there that can continue this cycle, because this is really what determines overall plant health, and goes back to the idea of what Dr. Hatfield talks about, is building soil health and biological fertilizers. So when we go to that information that we're receiving from Cornell and WSU, we have another tool in order to combat stress and environment. And that's really what we have to do to stabilize our revenue as a grower today. And we look at that as that's not only with our biological community, as we've been talking about the microbes, there's research coming out there on plant communication. Steve did a wonderful presentation quite a few years ago on just plant communication and how they communicate. He talked about the goldenrod, of how it was under attack. It was able to put out pheromones, if I remember correctly, and it was able to warn the other plants that it was under attack and to protect themselves from that attack. And we see that exact same thing happening with plants. This was some research done with tomatoes, where it's looking at the mycorrhizae fungi network of communicating between plants within that soil environment. And so, I mean, when we start to really dig into what all our biological community does, it's fascinating, the research and information and how important these guys are to be healthy as agriculture growers today, and exactly what Dr. Hatfield is talking about. And we're finding it's not only between plants, it also is not just tomato to tomato. It also the mycorrhizae can go from tomato to your cover crop. They infect each other. And I mean, this is just fascinating, the research that's coming out there, but it tells us how important that mycorrhizae is based on plant communication. We always look at mycorrhizae based on, well, it gives us more water and more phosphorus and a little more nutrient. It is so much more than that. So we have to really think about this as, what is the importance of mycorrhizae fungi? Well, it's more than just phosphorus and nutrients. It's overall plant health. It's communications of those plants. It's plants protections. The list just goes on. So with that, I want to talk a little bit about our microbes, and Steve has done quite a bit of research on these. I get a lot of questions on our biological packages, so I wanted to just talk about what is the foundation of what we build. And so most of our organisms are soil-based organisms. They're non-pathogenic, biosafety level 1. They're non-GMO. And the key here is they're individually grown. So each one of our microbe strains are individually grown, and then they're tested for counts, and then those individuals are combined together in order to build the product based off of function. And that's a lot of the research that Steve does, as he was kind of talking about, is how do these guys play together? You know, he was talking about your biofungicides, your bioinsecticides, your control products. They don't like to play nicely with everybody else. So it's putting everybody in a package that can play well together within that soil environment and create multiple functions. And with that, he also puts a complex media with full trace mineral profile to support that biological community. So when we tell you, yes, we still want you to use fish, we want you to use humates, we want you to use food sources for that biology, that's to build that population within that soil environment so it has a chance to grow. But they're basically sent with a sack lunch when they leave their office. We have within each of our mixes plant growth hormone producers. We have nitrogen fixers. We have organic matter digesters. We have carbon sequestration. We have phosphorus solubilizers, mycorrhizae helper bacteria, plant growth promoting rhizobacteria, nutrient reducing or redox organisms, a high level of pseudomonads, and the list goes on. So that's our base foundation of all of our products. And most of growers are familiar with our Spectrum line or our Biogenesis, Mycogenesis line. They're all built based off the base foundation. And we feel that's very important of how they play together. Bruce never wanted to take the single microbe approach. Later on, we're going to talk a little bit about our specific products, our Spectrum DS, our Spectrum PSB, our Spectrum NFB, and how they're different. And I'll get into that a little bit later, but the basis is all there. All the players are on the field. And so the benefit we get from this, as we all know, we get a healthy, balanced soil. We get digest and cycle organic matter. We improve soil structure with the glues, the glomulins, the polysaccharides that build structure to build that bigger sponge, as we're talking about, to do what Dr. Hatfield is talking about, where you can actually see soil aggregate change within the first 3 months of healthy biological systems. That's generally around the root zone where we have those root exudates going into that soil environment. And then as we don't do anything to disrupt that biological community, we start to really see that soil structure and soil health change over time. We can improve root depth by reducing compaction, which is a huge problem. More area to mine for soil fertility. Solubilize and retain nutrients, again, a bigger sponge. Decompose toxins. Things that we have used in the past that were in that soil environment now can be broken down by microbial activity to help make more nutrition available and eliminate plant stress based on what had been used. Increase water and nutrient cycling. We talked a little bit about that based off of the mycorrhizae fungi, in addition to what else they do, but basically that bigger sponge. And we talk about protect the plant from disease. You know, later on, I'm going to talk a little bit about biocontrol products and a little bit of information on those biocontrol products. But those biocontrol products are used to naturally protect the plant from disease. And I think a lot of that we can talk about goes into what Dr. White's research was and how that functioned. And sequester carbon. So a lot of information here, kind of building a platform. So why do we use these different products? And I want to just talk a little bit about our Spectrum Plus Myco and our Spectrum product. What is the difference? Well, the only difference between these two products, remember, they are all built based off of the basic platform. The Spectrum platform has basically the nitrogen fixers, the phosphate solubilizers. It has a little bit of everybody in it. And the Spectrum Plus Myco has all those players in it, plus the mycorrhizae fungi. And so I talked earlier about the benefits of mycorrhizae fungi and how important that is. So some of the things we need to think about when using the mycorrhizae fungi to make sure that it's effective. We need to make sure that we're using the right mycorrhizae fungi for the appropriate crop. So if we're doing blueberries, the endomycorrhizae that is normally used will not create a relationship with blueberries. They need the Iroquois mycorrhizae. There are some plants, about 5% of the plants out there, that do not create a relationship with mycorrhizae fungi. If you're planting those plants, there's no reason to use the mycorrhizae fungi unless you're going to maybe follow up with a crop that would create a relationship later, because those spores would still be within that soil profile. If we're looking at the pine trees and a few other plants that are the ectomycorrhizae, again, we need to make sure that we're using the proper mycorrhizae for that plant. About 85 to 89% of plants will create a relationship with endomycorrhizae. And then we have those few outliers out there that are a little bit different. So just make sure that we're using the proper product for the proper plant. And if they won't create a relationship, there's not really a necessary or a need to spend the extra money. We can just go with the basic biological package. But we need to remember that in order for that mycorrhizae to create a relationship with that plant, it needs to come in contact with the root. And that's why I really like to see in-furrow application is best. We want to make sure that we have that mycorrhizae spore where it can contact the root. So when that root has a need for phosphorus, it can create the relationship with the mycorrhizae fungi. Seed treatments are great. I talked about plant specific. One of the other things I want to talk about is when we're using mycorrhizae fungi, we need to remember it takes probably 2 or 3 weeks at least for that relationship to take part, okay, for that infection to happen. So if we are harvesting lettuce, for an example, and we know that we're going to be doing that in a 3 or 4 week period of time, does it really make much sense to be using mycorrhizae within that application? And I will leave that up to the grower to decide. If we're starting in the greenhouse and we've inoculated in the greenhouse and we're moving out to the field, then it probably makes some sense. But it doesn't happen overnight. We need to remember that with the mycorrhizae, it takes time. And again, parameters I give, I would just say 3 weeks is the time frame that we need to look at in order to create that relationship. And so when we talk about that, we talk about the benefits. This was some research that was done out of Ontario, Canada, and I just thought I would share it because it goes right back into what Dr. Hatfield talks about, and it goes into exactly what was done by WSU and Cornell based on environmental stress and instability in yield, and how important, in addition to communication and phosphorus and water holding capability, how important we're finding the mycorrhizae can be with the biological community. Remember, in all the biological packages, we also have mycorrhizae helper bacteria. We have everything in order to support this, for this to function as a system. Again, remember, it's a biological farm management system. It's a lot of things coming together in order to work. So with this research, they found that survival with mycopli mycorrhizae added to the plant, they had a 90% success rate. Without it, without the mycorrhizae, they had a 41%. That is a huge difference, almost a 50% difference in survival rate when we came under drought stress with these plants when using or not using, which is huge to a grower. 90% to 40%, I mean, at 41%, we're tilling and reseeding. At 90%, we can still be profitable. So very important to take a look at that.
When I think about some of the times, some of the stories you've told, and I think back to the one, those were Honeycrisp apples, weren't they? Where the root dip really it helped reduce stress. And again, a big part of this is reducing stress on the plant. If the plant is excessively stressed, its hormones basically go haywire, and it can't have normal cell development, it can't have normal root development, it can't do those normal things. So having some of this diversity, having some of these beneficial microbes can help reduce the stress, can't it, Dennis?
It can, and the story Steve is referring to, there was a trial we were doing on Honeycrisp apples in Colorado, and I got out a little bit late that afternoon. It was about 2 o'clock in the afternoon. It was a day very similar to what we're having here in the Pacific Northwest. About 90 degrees with about a 20-mile-an-hour, I call it oven wind. And when I got there, they were dipping the trees, and a guy was walking along the hole and just dropping the trees on top of the ground. And he was about 4 rows ahead of the people planting. So these trees were laying out in the sun with this wind, and their roots were just dry and crusty. And I pulled up with a grower because we were somewhere else looking, and I kind of lost it, I guess would be the best way to put it. But they were doing the exact same thing on the other side of the trial. They were just laying the trees out, and a couple guys were coming behind and planting them in the holes that they had already dug. The wonderful part about that, in the area that was root dipped with mycogenesis, we had a 99% success rate. We lost 1 tree. On the side where they did not get root dipped and were planted the exact same way, they had about a 50% strike rate. So they had 50% skips within that field profile. And I've seen that on apples. I've seen it on roses at the Broadmoor. I've seen it in numerous situations. I love the root dip scenario with the mycorrhizae. I see it in greenhouse settings, going out with a water wheel and planting, just reducing plant stress. It is tremendous. And so I want to talk a little bit just briefly about, I get a lot of questions. We've got the Spectrum PSB, the Spectrum DS, and the Spectrum our new product, Spectrum NFB, which I'll talk a little bit about later. It's been in trials here for a while. But we have to remember that all of these Spectrum products are built off the same platform. We do not want to use a single microbe approach or a single focus approach. We want all the other players there. So everybody else is there in each one of these Spectrum products. And then we have an emphasis on a function. So Spectrum PSB is basically an emphasis on phosphate solubilization. It's phosphate solubilizing bacteria. Spectrum DS is drought stress. It's sodium, high sodium soils. And then Spectrum NFB is nitrogen fixing bacteria. And we'll talk a little bit about how each of these products function. Well, I guess when to use each one of these products and when it makes kind of sense to pick one over the other. So with that being said, one of the things I'm seeing more and more of is the buildup of sodium in plants. And this is causes toxic levels, it causes stunted growth, it arrests cell development, it root pruning. We just don't see a healthy plant. Based on water quality, well quality, drought, all of the things that Cornell and WSU are talking about, and this would be on irrigated ground mainly, we're starting to see this problem. But we're also starting to see it in dryland farming based on agricultural practices, salt-based fertilizers, compost, all the different things that we can use. So it's not just for irrigated ground, it also can be in dryland farming. So we really have to pay attention to this because it can be devastating to the crop. This picture up here on the left, you can see this is the roots. This is spearmint, I believe, or the lack of roots, I guess I would say. You know, originally this was thought to have been a disease. It was sent in and tested and it came back negative. And I took a look at the field with the grower. We have not worked on this field yet and had taken the grower, the distributor had sent me pictures. And I looked at this and I asked if they had a tissue or a plant sap analysis, and the grower sent it in, and immediately I could identify what the problem was. And you can kind of see it based on the margins on this plant here in the middle. We're seeing the margins around the outside of the stress. This will eventually turn brown. We have poor cell development. We're losing more moisture than that plant's ability to take up. And, you know, this is very close to basically losing this crop. And this is irrigated ground. So when I started to take a look at this a little bit, I wanted to kind of figure out what was going on. And if you look at the middle bar here on the spearmint, this is the picture on the left is after the first cutting. This is the regrowth. And wasn't really noticed in the first crop grown, but it really started to express itself in the second crop. So if you look at this field history and we look at the tissue samples, our plant sap samples, if we look at May 27th and we go down to sodium, and we see sodium level was at 0.286. And we go up to the chart up above and we know that based on this analysis, they want to see your sodium level from a 0.02 to a 0.2. So the sodium level in May when they were growing that first crop was almost, well, it was exceeding the highest point or the maximum level that we would want to see this sodium level at. And then we look at it on June 10th and our sodium level is 0.547. So it doubled from May to June on the amount of sodium uptake in this plant. And we start to think about this. The grower asked me, do you want a soil analysis? I said, no, I want a water analysis because this is a very sandy soil. We are not going to retain a lot of sodium within that soil environment. And the soil test showed that we didn't have high levels of sodium in May. So this is coming from our irrigation water. And if you look up top here, not only are we having problems now with sodium, but if we look at our calcium, if we look at it in May 27th, our calcium levels were sufficient. We look at it in June and we're greatly deficient in calcium based off of this analysis. And so we really started to change the profile of how basically all of this worked. And actually the numbers are wrong on that chart that I made down there. I actually transposed numbers, sorry about that. That second number for June should actually be 1.93. So we can see how quickly that changes. And we see the same thing time and time again. And so that would be a perfect scenario for using the Spectrum DS product. That's what we would want to take a look at based on using DS. As you can see across here, our calcium is low, but our manganese and sodiums are through the roof. So we're blocking calcium uptake by excessive sodium. So when we look at another soil sample here, or tissue analysis, we can see here that I want to emphasize phosphorus deficiency and sodium deficiency, or excess. So we have a deficiency and an excess. And a lot of times when I talk about that, I always feel an excess causes greater problems than a deficiency. So in this case, I would look at, based on these trees and the time of year, this is basically July 21st, I want to get the sodium excess under control. So in this scenario, we have a deficiency of phosphorus, but we have an excess of sodium. So I want to use the DS first, maybe come in with the phosphorus application to address the phosphorus, and follow up later with the phosphorus PS, or the Spectrum PSB. That would also depend on taking a look at the soil analysis and seeing where our phosphorus levels were. So again, a lot of times we always say test, don't guess. The example I can give you in that sodium issue, a soil sample was not going to tell us that we had excessive salt. We had to have a tissue analysis to make that decision. And now we have a program and a system that we can use in order to combat the problem. Now, when I talk about the Spectrum DS, for an example here, for sodium remediation, I guess we would call it, or to eliminate stress onto the plant, again, it's a biological farm management system. In addition to the Spectrum DS, we want to add some humate to suck up, as Steve always says, create a bigger bucket, suck up some of that sodium. We want to put some kelp out there to basically, kelp is a great stress reducer. We want to put some calcium out there to address the calcium level and the block calcium. So all of these things need to go together as a program when we start to target something like this. So it's trying to decide which product to choose. A lot of times I tell people, if they have a question or they have issues, feel free to give the office a call, talk to Steve, talk to myself. We'll be more than happy to help. So a little bit, I'd love to go in even more on each one of these products, but I know it gets kind of boring, so I'm just trying to give some general information. Sorry about that, guys, but I get so many questions. I thought I would kind of try and put this in a nutshell of how to choose which product and why. This is a trial on our Spectrum NFB, our nitrogen fixing bacteria product. We have quite a few acres out in Kansas and Nebraska. We have some in Washington, and they're doing fantastic. And again, our emphasis, you'll see products out there with nitrogen fixing bacteria. They're only containing a few of the nitrogen fixers. They're not containing the entire package. And we want to see that entire package together. The two pictures here on the right are spring wheat. The one on the left is not wheat. I know that. You guys are probably thinking I was crazy. I'll talk about that picture in a minute. But the two pictures here on the right are spring wheat that we did this year. Normally, the application here is 80 to 120 pounds of nitrogen in order to grow this crop. To date, there's been 20 pounds of nitrogen in 2 foliar applications. I can tell you early spring this year, we were running out of nitrogen. The nitrogen fixers had not kicked in. But as temperatures warmed up and those nitrogen fixers begin to function, basically this wheat is caught up. We're going to go down and look at this wheat. I'm going to look at it again next Thursday, and we'll probably be harvesting this wheat in about 4 weeks, 3 or 4 weeks. Talking with the grower yesterday, as he says, this crop appears to continually put on yield. He's just been very, very impressed. So we'll kind of keep an eye on it. I'll update on where our yields came out. I do want to let you know this is a conventional grower. The reason he's kind of making the switch to this NFB product goes a little bit to what we're talking about, again, based on Dr. Hatfield's research in building soil health and what WSU is seeing based on drought. This is dryland wheat. And so this is a no-till grower. They have found that by their no-till system, the way they are doing it now, they are not making the strides in soil quality that they would like to make. And part of that is because we're burning up our carbon by using 80 or 100 pounds of nitrogen. We're not actually making the jumps that we would like to in building soil health and water holding capability, the bigger sponge, as Steve refers to. And the only way we're going to do that is to try and grow this same crop with better nutrition and only utilizing the nutrition that that plant needs, so we don't have excess where we're burning the soil carbon up, so we can actually build carbon within that soil environment. And then it goes back to the idea of what we're talking about, are root exudates and biomass, dried dead microbial biomass, and all of those things that come in when we actually get that biological system functioning, building carbon in that soil so we can have a higher microbial population within that soil environment. So this is a conventional grower using the nitrogen fixing bacteria. I've got a lot of organic guys that are also using it because it gives them another tool in their toolbox in order to grow a healthy crop. The picture here on your right is beans, and I don't know if you can see it, but the grower was sending me pictures of the nodulations that were actually happening on these beans early on. Very, very excited about that. And the reason I point that out, I couldn't fit it into this slide, so I put it up there with that one, is there's a lot of research out there that co-inoculation of the rhizobium and then also the beneficial bacteria increase nodulation and increase nitrogen usage within that plant and nitrogen availability. So they show here the Bacillus megaterium was what was used as the nitrogen fixer. That is one of the nitrogen fixers in our product. There's a lot more in our product. It's just, again, not a single strain approach. But what we're finding is even with legumes, it makes sense to use the nitrogen fixing bacteria because, as we say, it's a system. Everything functions better. As I talk about the mycorrhizae and the mycorrhizae helper bacteria, these all function together within that soil environment. It's very important that we have them all together and all the players are on the field.
Yeah, and I mean, that's definitely the case. The idea of mycorrhizae helper bacteria is exactly what I was thinking. I don't think there's a classification of rhizobia helper bacteria yet, but with research like this, it's a pretty strong indication that there probably should be. Bacillus megaterium is also a fantastic phosphate solubilizer. And it was, as we know, nitrogen fixation, as I talked about last time, is one of the most energetically expensive processes that happens in nature. So it uses a massive amount of energy. And if you remember, the energy source that we primarily use is ATP, adenosine triphosphate. So we've got to have lots of phosphorus available. Those are our little tiny batteries. So this Bacillus megaterium is helping feed the energetic supply, the energetic demand these rhizobia need. And incredibly, they saw a little over a 31% increase in the amount of nitrogen that was fixed when they added these other PGPR, when they added this Bacillus megaterium. So it's not a little bit that they're seeing. It can be a fairly large quantity. And again, these are systems that are designed to work together. The idea of that pheasant from the very beginning, we want to have diversity so they can work together.
You know, and when we think about that, this is not only good for this year's crop, but think of how this builds for our next year rotation crop. If we're going into corn or we're going into wheat, all of this free nitrogen that we're receiving within that soil environment. And these are the things that we're talking about, is building that farm system, that biological farm management system, to all function together. And so with that, you know, I want to talk a little bit about our,
I think you skipped one.
Oh, did I skip one?
Mm-hmm.
I did. I skipped mycogenesis. I'm just going to talk about this briefly. I call it roots, and more roots. Pictures up here, you can see these were cucumbers grown in a greenhouse in Montana. The grower sent me these pictures. This was the first year he had used the mycogenesis product. He'd been using other biological inoculums up into this point with mycorrhizae in them. And he's been doing this for a long time and had never seen roots like this on his plant. You can see it clear down in the bottom of the grow rig, not only on the surface, He was just really, really excited. His plants are doing fantastic. Unbelievable crop that he's carrying on this, just continually harvesting. So, you know, we have the mycogenesis product, we have the biogenesis product, and we have the rhizogenesis product. So just to give you guys a little bit of an idea about those three products, rhizogenesis is not organic, cannot be certified organic because of the polymers in it. If anybody out there, as Steve always says, knows an organic polymer, please let us know.
Yes, please.
But it's designed as our root dip. As Steve was talking about earlier, the root dip scenario on those Honeycrisp trees that survived the stress of the heat. Again, as I talked about in-furrow application with the mycorrhizae and the biology, this is where they want to be. So I cannot talk highly enough about our root dip scenario. If you're an organic grower, you use the mycogenesis as your root dip. And if you're ever dealing with a plant that does not create a relationship with mycorrhizae, fungi, you can use the biogenesis product. I mean, that in a nutshell is really it. Greenhouse, infield application, row crop, hemp, cannabis, tree fruit, all of them can benefit by the use of the mycogenesis, biogenesis, or rhizogenesis product. And again, it comes down to the base platform with a lot more in a food source to support it. That's really what the biogenesis product is all about.
Well, I think you mentioned it. I mean, seeing these pictures of roots was just, I mean, it's incredible. It's almost too much for it.
It's almost too much.
It's pretty unbelievable. And this is a grower that wasn't new to biological approaches. He was already using a different type of inoculum, and we've changed a little bit, or I guess Dennis, you've changed a little bit of what he's doing, and focused him a little bit different on those inputs. But this is somebody that was already used to biological farming, correct?
That is correct, and just been tickled pink. It's been a fun year. And so we get a lot of questions about which foliar to use and why. And in a nutshell, I look at the 5,000 for growth or vegetative. I look at the 1,000 for reproductive and fill. And I look at the organic 5,000 for all season inorganic. Now, the one thing I will talk about here just a little bit, if we are an organic grower, as you can see, we have a micro 5,000 and we have a micro 5,000 organic. Please be sure when we order, or your distributor orders for you, that we make sure that we get the micro 5,000 organic, that it has the OMRI listing, the WSDA on the label, just to ensure that we don't run into any problems with certifiers. So again, just make sure that if we are organic, we're making sure we receive the organic products. If you have any questions, call myself, call Jenny, call Amy, Steve, anybody in the office will be more than happy to help you. But that's generally in a nutshell how I look at it. For growth, the 5,000, reproductive, the 1,000. You know, I can't tell you, I had a grower call me the other day, and he was talking about pumpkins and a little bit too much nitrogen on the pumpkins. They weren't going to flower like we wanted them to. I've been very successful, if you're a conventional grower, to use a double rate of the PZ1000 to shift that plant a little bit more into reproductive rather than growth or vegetative. There's other things you can do also. I can tell a story of a essential oil. I believe this was Melissa, that was growing, and it was getting a little bit late in the season. They wanted it to go to flower so they could harvest it. He asked me what to do, and I recommended a double rate, and it shifted those plants so quickly that after you get past a certain level, you actually start losing oil production when they went to flower. So they had their hands full trying to keep up with how quickly the fields turned and getting the harvest done in time. So sometimes there's things that we can do to manipulate the plant. But again, I always say, let's eliminate the excesses so that we just grow a healthy plant and we don't have to worry about that. And I want to talk just a little bit about enzymes. Why enzymes? What do enzymes do? I get this question all the time. We have basically two enzymes product, the Pepzyme G, which is designed for the conventional grower, and the Nutrineed, which is organic certified, that can be used for conventional and or the organic grower. So in a nutshell, basically every enzyme has a specific function. That's really what I want to talk about within that soil environment. So when we're using enzymes, it's like a lock and a key. We have these enzymes, as you can see, we go up here, number one, we have a bond of a substrate. Basically, it fits directly into that enzyme. The enzyme creates a reaction, breaks apart that bond, and it splits it into two parts, usable glucose and fructose in this situation. So we're making it available in a state that the plant can use it. That's, in a nutshell, what enzymes do. They do this for the plant. They do this for the beneficial bacteria. So I always get a question, why are enzymes important? This is it right here. It's another piece of the puzzle.
Yeah, and enzymes, I mean, you can kind of think about them. They're basically like scissors. They're specific scissors for cutting a specific thing. But the beauty is these scissors can be used over and over again. So you might have one enzyme that's breaking down, an amylase enzyme, for example, that's breaking down starch. But it cannot, it's not just one starch that it's breaking down. It can reuse that enzyme. That cycle can happen thousands to tens of thousands of times. And as our microbes wake up, the last thing that we want them to have to do is create their own. Creating enzymes is creating protein. Protein requires nitrogen, and again, nitrogen fixation is hard. Nitrogen availability can be hard, so we want to supply the microbes with an easy food source as early as we possibly can to maximize their establishment and growth. And that's a big part of what these enzymes are. It's kind of like a prebiotic. It's going to help the bacteria and the beneficial organisms grow by partially digesting their food for them. It's kind of like a mama bird. You can think about the peptide products kind of like mama bird chewing it up for them. It gets them ready, and it means that the system uses less energy. Those bacteria use less energy to establish themselves.
And let's just talk about biofungicides. I get questions all the time, can we mix your products with biofungicides or bioinsecticides? And I guess the easy answer here is, if it is like a trichoderma or a Bavaria bassania, a fungal component, and we're mixing like products, yes, they can be tank mixed together. In fact, a lot of research that we're seeing out there from growers is when they mix their biofungicide with our Spectrum product, they're actually seeing an enhance in overall plant health. So we're going after the control of whatever the pathogen or the disease of the insects might be, but we're also increasing support to a stressed plant with this biological package. So it works hand in hand. They work very well together. If we're using some of these that are not a fungal or biological control product, we have to take a look at those on a case-by-case basis. Some of those actually can be detrimental when being tank mixed because they're designed to kill. So one of the things I do want to talk about, and Steve touched on a little bit though, is when we talk about these control products, if we follow a biological farm management system and we have healthy plants, we hope we never have to go after a control product. But in nature, sometimes that happens. The thing we don't like about taking the single microbe approach, for an example like the trichoderma, it wants to be dominant within that soil environment. It wants to control the environment. And so when we say it goes in there and controls the environment, it can suppress mycorrhizae fungi inoculation. It can suppress other fungals within that soil environment. It can reduce beneficial bacteria populations within that soil environment. So when we take that single microbe approach for a control product, which sometimes we will need them, we need to make sure we go in and balance back out that system. We need to do something to support that system so that we have all the players on the field. We get the control we need, but then we rebuild our system.
If everything is going happy, great, and well, you don't call in the military. But when you call in the military, there's usually going to be a little bit of cleanup afterward. And that's kind of what we're talking about, is as we apply these controls, there's going to be a little bit of wreckage. We've got to come back in and do the things Dennis talks about all the time. The program approach, adding the food, adding the support materials, adding those microbes in population densities and diversities that can kind of come back in and rebuild those populations. I really like that. It's a good way to think about it, Dennis.
Yeah. So what's new? I want to talk a little bit about what we're seeing more often here. This is a plant sap analysis. thing that is exciting for us to see is we are starting to see labs recommend plant growth promoting rhizobacteria, nitrogen fixing bacteria. So we see off a plant extract analysis, we're seeing we need some amino acid nitrogen, which is a good food source for the biology. We need calcium silicate. We need a little bit of humic and fulvic acid, and we need a max rate of plant growth promoting rhizobacteria and nitrogen fixers. We're seeing this in recommendations on soil. We're also starting to see it on recommendations of foliars. So we're starting to see that the understanding, not only in nutrient availability, we can have a deficiency, we can put it out in that soil environment. And just because we address the deficiency with a soil application does not mean it's available to the plant. So we dump more and more on, and we're seeing levels go higher and higher, although they're being tied up and not made available to that plant because we're lacking the digestive system. And what is really cool is some new information that's coming out of a lab. This is based off a complete soil report with DNA sequencing. And I'll keep you up to date on this, but this is a tool that we've been talking about needed to start coming around in order to see how is our soil functioning, not only based on nutrition, but also based on the biological community within that soil environment. So as we see down here on the bottom, the conclusion is the health value is very low. They found 3 relevant diseases that may be at risk for this crop. You have a very low resistance value. Aggressive management can be affecting your soil biostability. And nitrogen, phosphorus, and potassium nutrition values are low. And so as we see up here on the top, our health is very low based on resistance, which means we're susceptible to everything out there. And we're going to have to use a biocontrol product in order to grow the crop. And a lot of times when we get there, we've already reduced yield. Over here on the right, you say we've got our carbon pathway is really good, but we're lacking our nitrogen pathway. Our inorganic nitrogen cycling is low. Our inorganic phosphorus solubilization is low. Our potassium mobilization or solubilization is low. And so we see we're missing these digestive packages within that soil environment to make these things available. So it's really exciting to start seeing this information come out. As this move forwards, I will keep you guys posted with where this is going, when it's going to be available, cost associated with it, all of those types of things. But we're really excited to see this.
Well, and I kind of look at that like you talked about with the pheasants, Dennis. I mean, you go out in that field, you look around, you have an eye that's kind of trained to see what's going on. A lot of people don't. And the hope, what we've been hoping and waiting for, is analysis type that basically makes it so that we can see, have a better idea and physically see what's going on down there. And that's where some of these new tests are heading. That's the direction that we hope they're going to be in. And doing some genetic sequencing and profiling gives us a better view of potentially who's down there. I mean, there's even things on there like methanogenesis, methane production, which raises a few more questions in my mind. Why? What's going on there? Are we anaerobic? What are they doing? So some of these tests are incredible. And as you mentioned, it's also a strong indication that these analytical labs are recognizing the importance of having that biological diversity and being able to introduce it if need be.
Yeah, and it's based off of microbial function, based off of DNA sequencing. Like you say, it still doesn't give us the players on the field, but it gives us groups of players on the field. So we know whether they're out there or not. So really exciting. It's really taking that next step. And what I've always said is some of our soil health analysis really don't give us the information we need, where we're starting to see the tests do this. So it's the next frontier. It's really exciting to see. And it's something that we've talked about for 20, 30 years that needed to be available. The technology just wasn't there. So with that, I have talked plenty. I'm going to end with a pheasant flying across the beautiful landscape and go to questions.
We had one from Karen asking about the Spectrum NFB. Amy's response, basically Karen is asking when, And with Jenny out of the office, we're going to have Jenny check in to see. We're working on registration now. It's a product that, as Dennis has mentioned, is in trial. We like how things are going. Lights look green so far, and so we're hoping to have that product available soon. So keep checking back, and we'll keep filling everybody in on that.
Yeah, so a lot of it has to do with the registration product, getting it registering, getting the label approved. All those things, I'm probably in really big trouble for introducing it today.
Oh, probably. Oh, yeah.
But it's just very exciting. We're having wonderful results. So please be patient and just check in with this. And as soon as all the registration stuff is done, we'll certainly get back to you and probably send an email out to all the distributors and everybody and let them know kind of where we're at.
Again, you're probably going to get in trouble for that. You're promising all sorts of things this morning, Dennis.
I know. I'm working from home, so I'm away from the office, so I'm safe.
It's safe. You don't get the evil glares as easily from a distance, do you?
Amy may stop by on the way home from work and have a little chat with me.
If you don't hear from Dennis for a while, we know why.
I'm in trouble. I'm in the doghouse. Anyway, everybody, thank you very much. Hope there was some useful information here. If you have any questions, please email myself or Steve. Give me a call, and we'll look forward to chatting with you soon.
Well, and one thought that popped into my head, Dennis, that we had kind of intended to talk about and didn't. I know everybody wants to go. The topic of rhizophagy with Dr. White, we mentioned the biofungicides and how those organisms are used to help control. Something that we should have mentioned there is the research by Dr. White is also showing that some of these PGPR, these beneficials, will actually go in and inoculate the fungal pathogens. They'll go in and basically attack them, and they change how they perform. They can actually physically reduce their virulence. Virulence is how nasty the disease is. You know, one year you get a flu that, it's pretty annoying. Another year you get a flu that really knocks you down. That's the virulence level. So these beneficial microbes can go in with your fusarium, with your Phytophthora, and actually physically inoculate them and infect them and decrease how harmful they can be in the environment, which is pretty incredible. And again, it highlights how critical it is to have this diversity. Diversity is going to be the key to bringing back all of these functions. And without those organisms, you can't have the function. And that's, again, that genetic lab is kind of showing the same thing. We have to have this wide diversity of species to have a wide diversity of function.
And yeah, it's a really good point. Like we talk about there of the basically around the root, on the stem, in the flower, inside the seed.
Mm-hmm.
As we learn more and more and more about the importance and the communication and the mycorrhizae fungi, all the things we've talked about today, I mean, it just builds upon itself.
It's layers.
It's layers.
Yeah.
And, you know, the more I call this, rather than peel an onion, let's put the onion back together, layer by layer by layer. So we get this huge ball of microbial community back within that soil environment function the way that it was designed. Kind of like re-inoculating the pheasant and then just keeping it going. We got the initial population out there. Now we just supplement that population on a regular basis and keep that population happy.
Yeah, it's a catalyst to spark.
Yep.
Well, very cool. Let's see for next time. So this, Dennis touched on the products a little bit. Something that we could do if people find it interesting is go through kind of product by product. So if there are other topics that you'd like us to talk about, and I see here quorum sensing, I think would be a good topic. Yeah, thank you, Faith. I agree. I've talked about quorum sensing a little bit in the past, and that would be a very fun topic. Seed biome, another interesting topic. So yeah, thank you. Please keep those topics coming. We look forward to chatting with you. We're going to have another webinar as of right now in August.
Yep.
So we will talk to everybody here very soon. And thank you very much.
Thanks, everyone.